Hello, good afternoon, everyone. Let's get started. We will try to finish up metabolism today, and whatever we don't finish, we're gonna, finish on Wednesday, and hopefully that will give us some time to review for the test as well. Next Monday is Family Day, so there will be no class, and your test will be on next Wednesday. Okay, so let's do a little quick recap for the things that we learned
Vincent,
last
we
week.
have another test today, right?
Yes, you have a quiz today as well. Yeah, the quiz. Yeah, the 10% quiz, which is going to be available until midnight tonight.
Oh, okay, so the midterm is next Wednesday.
That's right. The test number 1 is next Wednesday. Yeah, which is 20%. Yeah, of your final grade.
Oof, okay.
Yeah, they never end, do they? But then you would have the break after that, right? You have 1 week off from school, so that's something to look forward to, maybe, perhaps.
Yeah, I
So,
that's it.
sorry,
that.
Vincent.
Yeah, go ahead.
So the test gonna be from like lecture 1 to 2.
Lecture 1 to lecture 5 plus the lab. That's gonna be the test. Okay, this is gonna be test number 1, 20%, and it's gonna be next Wednesday. I'll post the information online, after you guys finish the quiz today, because I don't want people to get confused, right? But in a nutshell, that's what it is.
So Vincent, I have a question. If you so it's about cells, right?
Sure. Yeah.
Okay, so in the study guide, I have a question that says let me see, let me go there really quickly. I have a question that says so the application questions,
Okay.
the second one.
Yep.
Yeah, the second one, yes.
Mm-hmm.
Yeah, so does this mean that every organs in our body have their own cells?
Yes. So all the organs are in fact made up of cells. So heart has heart cells, lungs have lung cells, and so on and so forth. And depending on the function of the organ, the proportion, the number of certain organelles might be more than others. So because the heart beats all the time, you would expect it to have more mitochondria than, say, from a liver cell. And liver plays more of a role in detoxifying your blood.
It has more of smooth ER.
Exactly.
I see. So that means that there is no one particular cell for our whole body. So just like cells, we have like cells in every part of us, right?
That's basically it. Yeah.
Okay, I see. Thank you.
Yeah, they are like the basic building blocks for life, if you would, like the basic unit that makes up, the entire organism. Okay, so let's recap quickly some of the things that we've done. We wrote down the chemical equation for cellular respiration. I'm just gonna do it in the chemical equation. Okay, you can look up the word equation from the notes last time. I know I have not posted yet, but I wanted to post it, when it's complete, right? And you guys were busy with the lab and studying for the quiz anyways last week, so it wouldn't have made much of a difference. So the chemical equation goes like this, right? C6H12O6, which, as you know, is glucose, which is going to react with 6 oxygen atoms. Hopefully by now you have learned how to balance equation in chemistry. That's going to produce 6 carbon dioxide plus 6 water molecule and up to 36 ATP. Up to 36 ATP. Now this is called a redox reaction because we are removing electron from the glucose. When we remove electron from glucose, loss of electron, what do we call this process? Is this reduction or is this oxidation?
Oxidation.
That is oxidation, very good. Okay, so it's glucose is oxidized into carbon dioxide, and those electrons are ultimately going to end up on oxygen. So gain of electron, say this is reduced. Right? Even though we write the reaction as a single reaction like this, I told you it's actually separated into different stages. And within each stage, there is going to be multiple steps taking place. So the four main stages in cellular respiration starts with glycolysis. Glyco is sugar. Lysis is to break it. You literally break the sugar in half, and that's the whole thing in glycolysis. Where does this take place? Anybody remember? You can have your notes next to you, right, to look it up.
Cytoplasm.
In the cytoplasm, that's correct. Yeah, so this is in the cytoplasm. And the key products include 2 net ATP. Okay. We also created 2 NADH. Remember, NADH are they are like shuttle surface, right? It's the I can write down here for you on the side in case you need a refresher. The NAD plus can become NADH once it picks up electrons, and then it could go back, right? When it releases the electron. Right? Same thing for the FAD, which it will become FADH2. So it picks up electron, it becomes the reduced form, and it carries these electrons to the electron transport chain, the last step, lets the electron off, and then it becomes this oxidized form again, the one that is empty, and it just cycles back and forth, right? So as we remove electrons from glucose in glycolysis, those electrons are going to be captured by NAD+, and they will become NADH. Okay. In addition, we will generate 2 pyruvate. Okay, so pyruvate, they are 3 carbon each. Okay, glucose has 6 carbon. After you split it into 2, they become pyruvate. Okay. I want to remind you about this concept of 2 net ATP. In our notes last class, I told you how glycolysis is broken down into 2 separate phase. We have the energy investing phase, where you will be, investing 2 ATP to energize the glucose to make it want to react. And then at the later steps, you're going to have the energy yielding phase, the energy producing phase, where you will release 4 ATP. So you invested 2, you create 4. When you subtract the difference, you will have 2 net ATP. Net means you take away what you have invested, right? Now I'm gonna put a star here. These ATP, these 2 ATPs are produced by substrate level phosphorylation. Look at the notes we wrote last time. I told you there are 2 primary methods of creating ATP, right? You can use substrate level phosphorylation, or you can do oxidative phosphorylation. And let me just bring the note up to remind you. In substrate level phosphorylation, you basically have an enzyme, and the enzyme is responsible for transferring a phosphate from a donor onto the ADP, which then creates ATP, right? This is not a very common method. It only makes about 10, 11% of all the ATPs that you are going to be creating. And then the other method is the oxidative phosphorylation, which we haven't really learned much about yet, but it involves the last step, the ETC. Right? So the ATP that are produced here in the glycolysis, the method that we use to produce them is the substrate level phosphorylation. The first method, the less common of the two ATP producing method. Are we okay? Any questions before we continue with our note? Okay, so let's just quickly fill in the other ones for now. The next step is pyruvate oxidation. And where does that take place?
Excuse me.
In the where, I'm sorry?
Mitochondrial
May I
matrix.
ask a question?
Oh, yes, sure, go ahead.
Yeah. Sometimes you said we have 4 steps, and I remember that you have said we have 2 stages for the.
Okay, yeah, I understand. Yeah. So, let me go here. The cellular respiration, like the whole equation, has 4 stages. Okay, stage 1, 2, 3, 4. In the first stage, in glycolysis, there are two separate steps. Okay, we have step 1 to 5, and then step 6 to 10, which separates into energy investing and energy yielding. So within each stage, there are multiple steps that are further broken down, but we didn't go into the details of each of the steps. Do you have a follow-up question on that?
So we have 2 stages for the
Glycolysis, yes. So you have
phase.
2 main phases, right? You call them phase. First phase is energy investing, and the second phase is energy yielding. Okay, but both of these happens in the first stage of cellular respiration. Is that better?
Okay.
All right. Where was I here? Sorry, so where does this happen again? The pyruvate oxidation? Which part of the mitochondria specifically?
The matrix.
The matrix, that's right. I have not told you what the key products are just yet. We'll do that later. Also happening in the mitochondria, in the matrix, is the Krebs cycle. And finally, we have the electron transport chain, right? The ETC. And that happens in the cristae. Right? So let's continue with our notes here. Let's continue with our notes. Okay. So let's revisit this diagram to make sure that we are okay with it. So far, we have the glucose right here, the C6H12O6, And in the energy investment phase, energy investing phase, doesn't matter, it means the same thing. We are going to be using 2 ATP. Right? So this is like minus 2 ATP. Right? And then in the energy yielding phase, harvesting phase, again, doesn't matter, same thing. We are going to be producing NADH. Right? This is gonna go to ETC, right? Goes to ETC. And we're gonna have plus 2 ATP, plus 2 ATP. And in the end, we have a net production of 2 net ATP. And more importantly, we have these pyruvates. So what are we gonna do with these pyruvates, right? So this is the stage 2. Stage 2. Pyruvate oxidation. So the pyruvate, which again is 3 carbon, produced in glycolysis will enter the mitochondrial matrix. Okay. And as the name implies, we're going to oxidize it more. Oxidize just means removing more electrons from it, right? Removing more electrons from it. So there are 3 things that's going to be happening in this pyruvate oxidation, excuse me. So here's the pyruvate, 3 carbons. Okay. So the first thing that's going to happen is we are going to be remove CO2. So how many carbon does it have now after you remove a carbon, after you remove a CO2? How many carbon does the molecule have now?
2.
Yeah, 2 carbons, right? Very straightforward, very good. We're gonna take more electron from it. So anytime we release electron, something has to pick it up. And in the case of this reaction, we're gonna have NAD plus again. So that's gonna come and pick up electrons, and it's gonna become NADH, right? Now, at this point, the molecule is a little bit out of chemical energy because it has gone through many reactions already. Right? We already produced some ATPs and some NADH along the way. Right now, the molecule is low in energy, and that's why we have to recharge it a little bit. Okay, and we can recharge it by adding a molecule, and the molecule is called coenzyme A. Coenzyme A. And the purpose of coenzyme A is to energize the molecule so that it's going to react further. So that it will react further. Okay. This is similar to the energy investment phase of glycolysis. You need to invest. You need to do more investment now. You need to invest more energy now in the form of this molecule called coenzyme A. And in the end, we get something called acetyl-CoA. Okay, I know these names are really weird, but you know, that's what they're called. Acetyl-CoA. Okay, so this one only has 2 carbons from the original glucose. All right. So this reaction actually happens twice. For each glucose. Because remember, in glycolysis, the glucose split into the pyruvate, right? So each of those pyruvate is going to undergo the exact same 3 steps. And now we have 2 molecules of acetyl-CoA. And all this is happening in the matrix. Okay? Not the splitting part, right? The splitting part happens in the cytoplasm. Okay, I'm going to pause.
So when you say matrix, it is in mitochondria, right?
Yeah, the matrix of the mitochondria, the innermost portion, right? Remember, this is the mitochondria. The matrix is right here, right? Okay. Any other questions? Okay, let's just do some quick poll questions just to see if you guys are with me. And these are very straightforward. I think they're relatively straightforward, but let's do them. What do you think? It shouldn't take this long. I only have 6 answers. Are the rest of you not able to see the poll, or what's the problem? Okay, that's fine. Well, thank you to the 6 people who participated. The answer is 2. Right? Net ATP is 2, because you're gonna subtract the 2 that you invested, right? That's why it's not 4. Let's try another one here. Okay, please participate if you are able to. This is just for me to quickly check to see if you are following. Okay. Correct answer is 3 molecules of carbon. We just roll it down, right? You see, pyruvate has 3 carbons. Glucose has 6. 6 divided by 2 gives you the 3. Okay, this one is a little bit tricky. Let's try that. Okay, so at the end of glycolysis, which of the molecule has the most chemical energy? Well, does anybody remember how many ATPs can each NADH produce?
3, right?
3, that's right. So this can make 3 more ATP. Carbon dioxide basically have no energy, okay? It's just released as waste product. Never mind the pyruvate right now, and this is just 1 ATP. Okay. So how many ATPs do you produce in total in the cellular respiration? If you consider the whole equation, how many ATPs are we expect to produce? I'm sorry, how many?
36.
36, that's right. 36 ATP, right? So by the end of glycolysis, we produce 2 ATPs, 2 of the 36, and we have some NADH, right? So they produce 3 ATP each. So, 3 times 2 is 6. So right now, we've only have 8 ATPs in total, right? Which means pyruvate is going to continue to produce the rest of the 36 ATPs, right? We still haven't got all those other ATPs yet. The pyruvate will still continue to react in step 2, step 3, step 4, right, to make more ATP, right? So the answer is C. Okay? It still has a lot of chemical energy, right? You can still generate a lot more NADH and so on and so forth. All right, so that is that. Let's move on to stage 3.
I have a question on this page, please, Vincent.
Yeah, sure.
Okay, so one glucose. No. The next one, the next slide. The notes.
This one?
The notes. No. The notes.
Oh,
the
the
class
notes. Okay, sorry.
Yeah.
Yeah.
Yeah. This one.
Mhm.
Okay, so this you said the pyruvate oxidation is like twice.
It happens twice for each glucose. Because one glucose makes 2 pyruvate, right?
Yeah.
So each of the pyruvate will remove the carbon dioxide. Each of them will produce the NADH, right? So
Okay, so do they all call like one after the other or at the same time?
it's same time.
Oh,
Yeah,
okay.
parallel. Yeah.
Okay. Thank you.
Yeah. All right. Stage 3 now. Stage 3. What is the name of stage 3? Well, it's the Krebs cycle. The Krebs cycle. Sometimes it's also called the citric acid cycle. Doesn't matter, both are okay. So what is happening? Well, this is a little bit confusing, but try to follow. The acetyl-CoA, which contains 2 of the original carbon, right? It's gonna come in to, I mean, it's still in the matrix, but then there is a 4-carbon molecule that was already in the matrix. Okay, it has a name. It's called oxaloacetate. Oxaloacetate. So they're gonna combine with each other. I'm sorry, what is it? Oh, okay. So they're gonna combine with each other.
Excuse me.
Yeah.
So this oxaloacetate, does it have anything to do with the cellular respiration, or it just exists there on its own?
It's already in the matrix. Yeah, it's already in the matrix.
Already there. It doesn't have anything to do with the glycolysis or anything.
It's nothing to do with glycolysis, nothing to do with pyruvate oxidation. It's already in the matrix and it's participating in the Krebs cycle.
Okay. So it's just there for the Krebs cycle?
That's right.
Okay.
So they are gonna combine with each other. 4 + 2 is 6. Okay, and you get citrate. I'm really sorry, but like I know it's a lot of things, but citrate and citric acid, they mean the same thing. Okay. For us, they mean the same thing. No, like, there is a difference, but like we don't have to know. Okay. But when they say citric acid cycle, they're talking about the citrate. Okay. In any case, what's gonna happen is there's gonna be like multiple steps that are happening in between. All right, like we're talking about 6, 7 steps, which we don't have to know. All right. I mean, I can show you very quickly. Okay. Here. That's all the steps that are happening in between. Okay. I'm not gonna make you memorize this. All right. So just know that there are many steps in between. What's important is this is called one turn of the cycle. Okay, one turn. You will be producing 3 NADH. These numbers are going to be important, huh? 1 FADH2, and then we're gonna make 1 ATP. Okay. And at the end of it all, when you have produced all these energy carriers and ATP, you will recreate the oxaloacetate. And then that's gonna be available to combine with another acetyl-CoA coming in, right? So this cycle just keeps on turning and turning, all right? So of course, for every glucose, this is gonna be happening twice. It's gonna be happening twice. And that's it for the Krebs cycle. Who just put like a crying emoji? Okay, let me put this into one continuous flow for you, and hopefully it'll make sense. Okay, so in the beginning, we have glucose, right? Oh, sorry, I forgot. There's also carbon dioxide here. Okay, 2 carbon dioxide here. All right, so let's just put this in a one continuous flow so that you can see exactly what's going on. We got some glucose, okay, which we will split into pyruvate. Okay, say the name with me. It's gonna help you remember. I mean, you don't have to unmute, okay? You just say it on your own, okay? So this is the 6 carbon, and then they split into 2 pyruvate, right? Now, what happens is we invested 2 ATP, right? ATP. This is like investing it. You paying ATP. But that's okay. That's okay because in the end we're gonna generate ATP. We're gonna generate ATP, and we're also gonna generate NADH. NADH. Okay, that's what is this process called again? Remind me.
Glycolysis.
Yes, glycolysis. That's what this is. Okay. Glycolysis. In the cytoplasm. Then the pyruvate is going to go into the matrix where we will do more reactions. Remember? More reactions. I know we just wrote this down like a moment ago, but that's okay. Let's do it again. So we're gonna remove a carbon dioxide. Remove carbon dioxide. We're gonna get more NADH. Where will the NADH go? They will take the high energy electron and go to the ETC, right? And then we have the coenzyme A. Coenzyme A to recharge the molecule so that it's gonna want to react again. Okay? And at the end, what do we get? This weird thing called the acetyl-CoA. Acetyl-CoA. How many carbons, guys? Tell me.
2.
2. Very good. Very good. And what do we call this thing here?
Pyruvate oxidation.
There you go. Pyruvate oxidation, right? Not too bad, isn't it? I'm trying to be encouraging here, okay? I know it's a lot. I honestly do. Okay. But we are doing good. Hopefully you're doing good as well. Then we're in the matrix. We continue to react in the matrix. This thing's gonna do the cycle, the spin, right? And then we're gonna get a bunch of stuff. We'll get 3 NADH, we'll get 1 FADH2. This is not the popular carrier. We only get it once, okay? And get some more. Get 1 ATP here. Okay. 3 NADH, 1 FADH2, and then ATP, right? And then it's just a cycle that keeps on going.
What
As
about
lo-
the CO2?
Oh yeah, I forgot the CO2 again, eh? Thank you. How many?
Two.
That's right. 2 CO2 here, 2 CO2 here, right? Thank you. So look. Sorry. Let's write this name. What is this thing called again?
Krebs cycle.
Krebs cycle. It's a name, it's a person's name, okay? Krebs cycle. Forgot his full name. Doesn't matter. Okay, so what happens? Well, we started with the glucose. Okay, but what happens in the end to the glucose? Well, you've broken it down into carbon dioxide. 1, 2, 3, 4, 5, 6. Look at the overall equation. How many carbon dioxide do we produce at the end on the right side? 6. That's where they came from. Okay. And we also have a bunch of energy carrier. We have a bunch of NADH. Okay, some more NADH here. Right? We have some FADH2. Right? And then some ATP. ATP here, ATP.
Vincent.
Yeah.
So in the first one, the second one is the pyruvate oxidation. For these ATPs, the ones in purple, the ATPs, do we move them to the pyruvate oxidation to, like, do we use them?
Oh, so once you make the ATP, you can use it right away, right? You can use it for, like, movement, for transport, for chemical reactions, right? Whatever you need them for, you can use them right away.
Okay. So are they being used for the oxidation at all? Like, are we moving them down?
No. They are good to go. You can use them for whatever purpose. It's done, right? So my point here, I guess, is of the 36 ATPs, which we're gonna produce, right? We've only made 4 of them right now, right? And if you have a calculator, you go like, actually, you might not need a calculator. 4 over 36, that's 1 over 9, right? Which is 11.1111, right? So about 11%, right? These are the ATPs that are created by substrate level phosphorylation. You know, the one with the enzyme takes it from a donor and put it onto the ATP. That's how these ATPs are made. But like I said, not the common method. Only 4 ATPs are made this way. Okay. So, are we together, guys? Any questions? All right. So what am I gonna ask you on the test, right? Let's do a few more poll questions to find out. You should read the notes yourself, right? Okay. It's basically what I told you. I don't want to read it to you from the PowerPoint. Then you can do that on your own. And as I said, it was harder to learn this from the PowerPoint than it is from taking notes. That's why I'm writing notes for you. This is a summary, right? We can fill it in together later on. But I really want to go through this. Okay, go ahead. Okay, has been a minute, but I only have like 4 or 5, 6 now, 7. Okay. You guys just wait until the last minute before you lock in your answers. Okay, okay. Let's take a look then. First one. NAD plus has more chemical energy than NADH. Well, that is not true. Remember, NAD plus is the empty taxi. It hasn't picked up the passenger yet. It hasn't picked up the high energy electron yet. So why would it have more energy than NADH? Right? NADH is the one that's energized. It's the one that picked up electron. Okay, so that's not true. NADH has more chemical energy than FADH2. That's true. How many ATP can NADH produce?
3,
3,
And what about FADH2?
2.
2.
That's right. That's why NADH has more energy, right? Next, NAD plus can be oxidized into NADH. Nope, can be reduced. Reduced means picking up electrons. Glycolysis can still take place without NADH. Nope. Without the NADH, nothing is available to pick up the electrons, right? To take them to ETC, right? Okay. So if you don't have anything to pick up the electron will stay with the glucose, and glycolysis cannot take place. All right, this one. I mean, we did something similar last time, but let's see if you remember how to do this. Okay, you will see these type of questions on the test. Okay. Yeah, all of you who participated got the right answer. All you have to do is multiply this by 3 and multiply this by 2, and then you add them up, right? Each NADH makes 3, so 4 of them makes 12. Each FADH makes 2. 3 times 2 makes you 6. Add them together, you get 18. That's an easy question. This is the harder one. Okay? This is the harder one. Let's see if any of you gets it. Okay, let's take a look. Now, it says 5 turns of the Krebs cycle, right? So look at the notes we just took. 1 turn is 1 circle, okay? 1 circle. So 1 circle, we have 3 NADH, 2 FADH, sorry, 1 FADH2, and 1 ATP, right? So how many ATPs do we make from the 3 NADH? Well,
Nine.
that's right. Times 3, we get 9. This one we times 2, and this is just 1. So 9 plus 2 plus 1 is 12. Thank you. And that's for one turn, right? You turning it 5 times. So multiply this by 5. We get 60. Okay. That's all. All right, now comes the super confusing part. The ETC. Let's go through it together. Electron transport chain. Hey, some good news for you, maybe. There will be no written questions for the test. Everything is just going to be multiple choice. So maybe that makes you happier or not. I don't know. Depends what you like. All right. So this is the mitochondria. And we're going to be like zooming in here. Going to be zooming in. If this is your paper, okay, you want to take up the whole page. Okay, don't just do a tiny corner here. All right. So here we go. Like that. That's just the mitochondria, but zoomed in. Let's put some labels here. This is the cytoplasm. What is this space here, huh? Can you see? What would this space be?
The matrix.
Yep, totally the matrix. And here?
Outer membrane.
I mean, this is the outer membrane. This line here, that's the outer membrane.
Okay, maybe inner membrane then.
Oh, sorry. Yeah, this is the inner membrane though. The inner membrane. Yeah. So what is the space between the two membranes? Does anyone remember?
Intramembrane space.
That's right, the intermembrane space. Good try though, Amita. Intermembrane space. Okay, the cristae will be these things, right? These little wavy things, those are the cristae. So just wanted you guys to understand what we are looking at in relation to the mitochondria and everything. So this is where the Krebs cycle was happening, along with the pyruvate oxidation and all that stuff. All right, moving on. Within the cristae, we're gonna have a bunch of these proteins. Okay? You're gonna have a lot of them. Okay? I'm just only gonna draw 2, so it's not gonna be like too cluttered. But this whole thing, this is the ETC, the electron transport chain, which is basically a series, a group. Okay, we say a group. Oh, sorry, the program is a little bit laggy today. I'm not sure why. A group of membrane proteins. found in the crystal. Each subsequent protein in the ETC has a higher electronegativity than the previous one, causing electrons to flow down the ETC. Just copy it first. I will explain what all these things mean in a second. Electronegativity means an attraction for electron. So let's just call them, you don't have to write this, but let's just call them 1, 2, 3. When I put an electron onto number 1, the electron will get attracted to number 2, which would then get attracted to number 3, because 2 has a stronger attraction for electron than number 1, and 3 has a stronger attraction for electron than number 2. So it just pulls the electron to the next one, to the next one, to the next one. If this concept of electron confuse you, think of 1, 2, 3 as magnets. Number 2 is a stronger magnet than 1, and number 3 is a stronger magnet than 2. When you put a metal object at number 1, it gets pulled to number 2, and then it goes pulled to number 3, because the strength of the magnet increases as we go down. Do you understand? Instead of thinking in terms of magnet, same thing for the electrons. The strength of the pull on the electron increases as we go down. So 1 passes the electron onto 2, 2 passes the electron onto 3, and so on and so forth. Okay, I'll pause for questions.
Yeah, so if that means if 3 I don't quite understand the 1, 2, 3 part.
Okay,
Yeah,
I'm
so
just
if
saying.
all of them have electrons, right?
Say that one more time. All of them have what?
Do they all have like electrons? Like, are they all looking to gain
they are all
electrons?
empty right now. They're all
Oh,
empty right
okay.
now. Yeah, but
Okay.
when we put the electron on the first one, that electron will get sucked toward number 2, and then it will get sucked toward number 3.
Number 3. Oh, okay.
Yeah,
Okay.
because each one in the chain has a stronger attraction, higher electronegativity, means stronger attraction for the electron than the previous one. Right. So number 2 will pull the electron from 1, and then number 3 will pull the electron from number 2.
Oh, okay. Thank you.
Yeah. Any other questions? Okay, it's only gonna get more confusing. All right. So please, if something's not making sense right now, it's a good time to ask. All right. So here we go. All those NADH and FADH2 that we produce, right? They collected the electron from glycolysis, pyruvate oxidation, and Krebs cycle. They will shuttle them all the way to the ETC. So here is NADH. It's gonna go to the first protein in the ETC. It's gonna let the electron get off. Right, so we're gonna have the electron right here. I don't know if you can see that color. Let's just change to the orange and see. Maybe that's a little bit better. Okay, so that's the electron. It gets off. After the electron gets off, the NADH becomes NAD plus again, right? The empty version. Where is this guy gonna go? Where will the NAD plus go?
To the second one?
to the second one here, you mean?
Yes.
Good try, not quite. Where do you think the NAD plus go? The electron just got off, right? What does the NAD
The
plus go?
pyruvates?
To where, I'm sorry?
The pyruvates?
Be more specific maybe, so I understand that you actually mean what you mean. What do you mean to pyruvate?
It's going to get oxidized.
Okay, I see. Not quite, not quite. Anyone else? Where will this go? Okay. Goes back to stage 1, 2, and 3. Why? To do what? What is it gonna do in stage 1, 2, and 3? To pick up more electrons, right? Remember, the NAD plus is like the taxi, right? Drops them off at the ETC. Now it goes back to 1, 2, 3, pick up more electrons, and then come back again. Just goes back and forth. Does that make sense? All right. So let's take a look at this electron. As the electron gets passed down the ETC, right? And I already explained to you why it's going to go down the ETC, because, you know, the next protein has a stronger pole, right? Higher electronegativity, right? As the electron it's being passed down the ETC, the energy from the electron is going to be used to transport H plus from the matrix into the intermembrane space. Okay, I'm going to write it here. Energy from electron is used to pump H plus from matrix into intermembrane space. Okay. So you're gonna end up having a lot of H plus here. Now you will still have some H plus here. Now you might wonder, hey, where does the H plus come from? Well, one source of it is the NADH. When the NADH becomes NAD plus again, the H kind of detached, right? And it's going to be in the matrix. So we're going to pump it in here, and this is going to have high concentration of H plus. And inside is going to have low concentration of H plus. Right? Any comments? I'm just really worried because I can't see your facial expression. So there's no way for me to tell whether you actually are with me or not. That's why I keep on asking you every 5 minutes if you have any questions. So all those high energy electrons, we use their energy to pump the H plus into the intermembrane space and create a high concentration. Now, you learn from me in lecture 4, things go from high concentration to low concentration on their own, right? So all these H plus that are stuck in the intermembrane space, they want to come back in. They want to go back into the matrix. But they can't, because the inner membrane is blocking them, right? The inner membrane is like a phospholipid bilayer. It prevents charged particle from going through, right? So even though we have all these H plus in the intermembrane space that wants to come in, it cannot. So this scenario, don't draw this, but I'm trying to, use an analogy to help you understand. This scenario is like, can I choose my black pen here? It's like building a dam, okay? Like you block off all, oh, that is some dirty water. You block off the water. Okay? You block off the water, and it's not able to flow because the dam is blocking it. So you create like an alternate path for the water to flow through, but you're gonna put a turbine here. So as the water goes through the turbine and come out, right, it turns the turbine, and in real life, this creates electricity, right? Understand that? But in order to do that, you got to dam the water first. Right? You got to block it from flowing down on its own. You force it to go through a different path, right? So that's exactly what's going on here. The dam is like the inner membrane of the mitochondria, right? And then the water, it's like the H plus. And then the turbine is this thing that looks like a doorknob. Okay, every single textbook makes it look like a knob like this. This thing is called ATPase, or sometimes you read it online, they call it ATP synthase. It's the same thing, okay? That doesn't matter which one you use. I'm just telling you in case you run into it online when you're reading it. Okay, so all these H plus, the only way for them to come back into the matrix, and they do want to come in, is through this special turbine, the doorknob protein, the ATPase. As the H plus comes in, it literally spin the ATPase, and then you produce ATP. Lots and lots of ATP. Okay. So this process of ATP production accounts for the majority of the ATP produced in cellular respiration, and it's called oxidative phosphorylation, right? Hold on, I'll come back to this. Right. Oxidative phosphorylation, right here, number 2, as opposed to substrate level phosphorylation. Right? So most of the ATP, 90% of the ATP is produced using this method. We are not done with this picture, but any questions? Okay. Well, we
Vincent.
yeah, go
Okay,
ahead.
so once this, let's say, turbine thing spins, that's when ATPs are is being produced, right?
That's right.
Okay. So
Just like as the water goes through the turbine turns and creates electricity, right?
yeah.
Yeah.
So once the this ATP synthase thing spins, ATPs are produced. So does this mean this spinning thing is what is called the oxidative phosphorylation?
Oh, not quite. But this
okay.
whole process of, creating the H plus concentration, and then they flow back in, and then we create ATP, this is called oxidative phosphorylation, right?
Okay.
So why, right? Phosphorylation means to put a P onto ADP, right? But
Yes.
what's driving this process? Well, ultimately is oxidative reaction. It's because we pull the electron from the glucose, right? That's what the oxidation is, right? We pull the electron from the glucose, and then we save all those electrons until up to this point, and we use those electrons to create a gradient and to create the ATP. That's why it's called oxidative phosphorylation.
The oxidation is when they lose electrons, right?
That's right. So this whole process is driven by the fact that glucose has been losing electron.
Okay.
And then we are capturing those electrons and then using those energy to make the ATP, right?
Okay.
Yeah. Now, what's missing in this picture, you might have noticed, is the FADH2. Oh, that's a terrible color. Let me see if I can find something that shows up a little bit better. I want to use a different color, though, from the NADH. But whatever. I guess I'll just use pink here. So that's the FADH2. Instead of depositing the electron on the first protein, they're doing it on the second protein. Okay, so the electron will still get passed down the ATC, but because it passed through fewer numbers of protein, is not able to pump as many H plus. It's still going to pump H plus, but just not as many. And as a result, when those H plus comes back in, it generates less ATP. Right? That's why NADH can create 3 ATP, but FADH2 can only create 2 ATP. So this FAD goes back to stage 3. Right? Only the Krebs cycle uses FAD to pick up more electron.
Okay, hold on. This is a lot. Okay.
It is, yeah.
Okay, so we know that NADH and this FADH are electron carriers.
Yes, they are.
Okay. Now this NADH, this NADH is coming with its own electrons. That's why it is this NAD plus is coming with its own electrons. That's why it's NADH.
Sorry, one more time. The last part, I didn't quite catch all of it.
Okay, I'm saying that this NAD plus is coming with its own electrons. That is why it is NADH.
Okay, so NAD plus, the difference between the plus and the H version for us, okay, it's whether it has the electron on it or not.
Yeah.
Yeah, so the NAD plus is the one that's without the electron. It's empty.
It's empty. Oh, okay. Okay, so the NADH is the one with the electrons.
That's right.
Okay, so now if it has given the electrons to these proteins or in the cristae, they are the one producing the H plus.
That's right. So like after the electron gets off from the NADH, then it becomes the NAD plus again.
Yeah. Okay.
It's empty again. It's ready to go back to glycolysis or whatever, right? To pick up more electrons.
Okay. Now the second carrier, which is the FAD.
Yeah, FAD. Yeah.
Yeah. So it's also coming with its own electrons, right?
That's right.
Okay. So where is it depositing its own? Is it also in the proteins?
It's also depositing on the protein, but the difference is rather than doing it at the first protein, the FADH2 is doing it like at a later time, at like a second protein in this case, right? So that's why. Okay, this is not entirely accurate, but you can think of it like this. In my drawing, NADH puts the electron on the first one, right? So therefore, it's able to pump 3 H plus, right? Right? Each protein pump 1. You can think of it like that, right?
Right. Okay.
And then each of these H plus comes back through the ATPase and make 3 ATP in total, right?
Okay.
The FADH2, it enters at a later point, so it's only able to pump like 2 H pluses.
Okay.
And when they come back, you only can make 2 ATP.
Okay. Thank you.
Yeah, that's good. All right. I know we are well past the break point, but let's just push through here and try to finish the story first. All right. Now, I am gonna give you a story here to help you understand this next part. When I take my kids to the park, there is something that really annoys me, okay? I mean, they're older now, so like they can take care of it themselves, but like when they were younger, okay? There is always that kid that, this is a slide, by the way, okay? There's always that kid that goes down the slide at the bottom and kind of stay there, right? They're not getting off. They're just staying there, lying there, not sure what they're doing. And I always tell my kid, like, don't go down until the kid at the bottom got off. Otherwise, you're going to run into the kid's head, right? And so every now and then there's that kid that gets stuck at the bottom, right? And then what happens? What happens is the next person cannot go, right? And then the parents have to come along, okay, and pull their kid off the slide so that the next person can go. Okay? My point is, if there's something stuck at the bottom, the next one cannot go, right? And that's what's happening with the ETC. I'm going to draw a new picture because the other one, it's like, I don't know about yours, but mine is totally out of space already. Yeah, so I'm going to draw a new one here. And I lost my favorite purple color right there. Okay, so that's the protein 1, protein 2, protein 3. Okay, I'm just simplifying things here, okay? Don't be surprised if you watch something online and there are more proteins, okay? So we have the electron. We have the electron here. The electron's gonna hop on and then it's gonna get passed down. So that's like the kid sliding down the slide. But it doesn't want to get off. And if it doesn't get off, we're gonna have a problem because when the NADH, the next NADH comes and want to let the electron off, it can't until the previous electron is pulled off the chain. Okay, until like the parent comes and pull the kid off. Right? Otherwise, the next one cannot go, right? So who's the parent? Oxygen. Right? If you have been paying attention along, you will notice we have not used oxygen yet, have we? Right? So the oxygen is called the final electron scepter. Its job is to pull the electron off. Okay, and what does it become when it pulls the electron off? Pulls the electron off the ETC and becomes what? You know this. I told you already. What does the oxygen become when it picks up the electron?
Reduced?
Yes, it is reduced, but into what? That's a good start. Let's go back all the way here. Look at this. Yes, Mary, what is it?
Pyruvate?
Into what? Oh, did you say pyruvate? Not pyruvate. Good try though. Look at this reaction, guys. What is oxygen gonna get reduced into?
Water and
Water, right?
Yeah.
Water.
oh,
Okay. So let's go back to here. Everything is related and becomes H2O. All right. So where do these electrons came from? They came from glucose. And after, passing through all these steps with the electron carrier and the ETC, ultimately, eventually, they all end up on the oxygen, right? And when the oxygen picks them up, they get reduced and they become water. Make sense? That's the ETC. That's the electron transport chain. All right.
okay, so this means that without oxygen to pull all the electrons that the NADH and the FADH have brought to the proteins, nothing is going to happen. Like everything is just like a waste of time.
That's right. That
Okay.
is exactly what I was going to say next, okay? You took the words right out of my mouth. Without oxygen, if we don't breathe, okay? Without oxygen, why do we die? Okay, the reason we die is because without oxygen, the electrons are not being pulled from the ETC. The NADH and the FADH2 has nowhere to put the electrons. And the electrons are not flowing from the ETC, right? Because it's clogged up with this thing that's not getting off, okay? With the kid, right? Like the slide situation. If electrons are not flowing down the ETC, we cannot produce what? What are we not producing when the electrons are not flowing?
Water?
Yeah, but water is not the key thing here, right?
Oxygen.
No, oxygen is what we breathe in. Why are we doing this, guys? Why are we doing the ETC? What's the ultimate purpose? It's making water the ultimate purpose? What's the purpose? Oh, I feel like I totally failed you guys.
Cellular respiration.
Yeah, why are we doing cellular respiration, guys? What molecule do we need?
Glucose?
Energy. ATP.
ATP, yes.
Ah,
Are the
okay.
rest of you with me? Come on, right? Like, what is the purpose of this? The purpose of this is to create the 36 ATP. Why? So our hearts can keep on pumping, our brain can keep on
Oh.
working, right?
Oh, okay,
Okay, yeah, like all the muscle
okay.
can move, right? That's the whole point of this. Okay, so we produce ATP by burning the glucose. Okay, and doing this whole ETC situation. But if you don't breathe, if you don't have oxygen, like, I mean, if we don't eat, we also die, just not as quickly, right? They say there's the 3-3-3 rule, right? I don't know if you heard about it. A person can go without breathing for 3 minutes before dying, 3 days without water, and then 3 weeks without food, right? That's like the 3-3-3 rule, right? So if you don't eat, eventually you will have no glucose and you die. If you don't breathe, you're gonna die more immediately, right? And I was trying to explain to you, without oxygen, there is no final electron acceptor available to remove the electron from the ETC. If you don't remove the electron from the ETC is clogged up with electron. The electrons don't flow anymore. We cannot create the H+, we cannot produce ATP. Without ATP, nothing happens. We die. Okay?
Excuse me, even still I didn't get the oxygen where it come from.
We
The
breathe it in, right?
oxygen.
We breathe it in.
Yes, this oxygen, the one you already.
Yeah, this one right here.
Yes.
That's the one we breathe in. Yeah.
Yeah.
So when you breathe it in, it goes into your blood, and
Mhm.
all your cells are connected to the blood, right? Through the blood vessels. And it goes into the cell where you will use it. Right? And all the carbon dioxide that are produced along the way, we breathe them out, right? Okay. Anything else? The rest of the class?
Please go back up. I need to like write something. Yeah, here. Thank you.
Yeah. Meanwhile, ask something. Okay. I know this is very confusing. Okay. So I'm just gonna go through one more thing with you. Actually, you know what? Hold on. Let me see. Let me see what's going on here. I think you know what? Let's just stop here. It's already a lot. Okay. So what you should do though between now and next class is maybe redraw this thing in a way that you understand it. Okay. Maybe redraw it. Based on your understanding of what's going on. Right? Maybe reorganize some of the writing and the drawing. And when we come back on Wednesday, we will try to do the calculations. We will find out where does the 36 ATP come from. Okay, so far I've only accounted for 4 of them, right? There are the 2 from glycolysis and then 2 from the Krebs cycle. The rest of them, we have to figure it out. Okay, we know it comes from the oxidative phosphorylation, but we got to do the math to show that it's 36, right? And I will also tell you what happens when our body is exposed to different kind of poison that would disrupt this process. You guys might have heard of cyanide, right? Cyanide is very poisonous. It kills you in very small dose. It turns out it interferes with this ATP production process. Yeah, so we're going to look at that. I'll see if we have time to do a test review. If not, that's okay. I have recordings from previous years for the test review. I will have a review sheet for you to try as well. But that's it for today, okay? Go through this stuff, do your quiz, and if you have questions, please email me. Okay, have a good one, guys. Take it easy. Bye-bye.
Thank you.